cells are unquestionably the most important cell type in our body. Since the moment we are conceived to the moment we die, they participate in very vital processes, such as processes that are responsible for body development and growth and tissue repair due to damage and disease. So it's not surprising that since their discovery in 1963 by two Canadian scientists, these cells have been explored for therapeutic applications. Just 5 years after their discovery, in 1968, an American doctor was able to perform the very first successful transplant. He took blood stem cells from a healthy donor and transplanted them into a patient. Many years later, currently, there are 7,000 clinical trials that are performed worldwide to explore the therapeutic uses of stem cells. However, just like 50 years ago, blood stem cells remain the only stem cell type that is FDA approved to be used in humans in North America. What is the delay? What is the reason for the delay in therapy development? Well, one of the reasons is that stem cells have to be derived from healthy donors, sometimes using very invasive and painful procedures. Secondly, stem cells are a very rare cell population, and therefore, in order to get enough cells for a transplant, they need to be expanded outside of the human body, which usually leads to their accelerated aging and loss of function. In our laboratory, in Radiobiology and Health Branch at Canadian Nuclear Laboratories, we are looking at the effects of low doses of radiation on function of human stem cells. For example, stem cells that form cartilage, which is a soft tissue that keeps our joints lubricated and fully functional. So in our studies, we have discovered that if we take freshly isolated stem cells, they very readily form cartilage. However, if we take the same cells and expand them for about 60 days, they age in culture and lose their ability to properly form cartilage. If we take young cells and irradiate them with low doses of radiation and expand them again for 60 days, these cells continue to be functional and actually outperform the young cells. Thus, we have concluded that low doses of radiation result in delayed aging of human stem cells. This work has now been patented. So how we discovered the fountain of youth, we don't know yet. We still need to follow up with the future studies. Thank you.